Welcome to the problem-solving set on mitosis and the cell cycle. Question 1 states that a pea plant cell with 18 chromosomes undergoes mitosis. How many daughter cells are formed, and what is the chromosome number in each cell? So remember that mitosis yields two daughter cells that are identical. So for mitosis, you have one cell, just say number, and it yields two identical daughter cells with the same number. So therefore, two daughter cells, and they each have 18 chromosomes in each. For B, if the same cells undergoes meiosis, how many cells are formed, and what is the chromosome number in each cell? So for meiosis, this is where the formation of gametes happens, and that is where one cell is split into four cells that have half the chromosome number. So you get four daughter cells, and they each have 9 chromosomes. And remember that gametes are not all identical, and that is the basis of recombination and sexual reproduction. Question 2 states that dogs have 78 chromosomes in each somatic cell. So dogs are diploid organisms, so that means that 2n equals 78. So A, how many chromosomes does a puppy inherit from the male parental dog? When a diploid organism makes gametes, they make gametes that are n, which is half the number of their chromosome number. And they get that amount of chromosomes from each parent. So the amount of chromosomes that the puppy inherited from the male parental dog would be 78 divided by 2, which is 39. For B, how many chromosomes are present in the canine egg cell? Again, when you make gametes, you have the number of chromosomes, so again, this is 39. Assuming dogs have the same number of sex chromosomes as humans, how many autosomes are present in each canine somatic cell? So humans have 2 sex chromosomes. And an autosome is a chromosome, it's every other chromosome besides the sex chromosome, so all except sex chromosome. Okay, so if humans have 2 and it's saying assume dogs have as many as humans, how many autosomes are present in each canine somatic or body cell? So this is body cell. So for it says up here, it says that they have 78 chromosomes in each somatic. So 78 minus the 2 sex chromosomes equals 76 autosomes. And for D, it says under the same assumption that they have 2 sex chromosomes, how many autosomes would be present in a canine egg cell? So if they have 76 autosomes in a somatic cell, that means that for the egg cell or a gamete cell, they have half this number, so that would be 38. Question 3. A male gamete and a female gamete with a haploid number of 5 fuse to form a diploid cell that develops into a multicellular individual. If this individual later undergoes meiosis, what is the probability that a gamete formed will contain a set of chromosomes, all of which came from the original male gamete? Assume no recombination. So let's just draw out 4 different shapes for chromosomes, and we'll start with the male, and we'll label his chromosomes in blue. So we have 1, 2, 3, 4, and 5. And then the females will label in red. So we have 1, 2, 3, 4, and 5. Now if these 2 gametes fuse, so remember that these are n equal 5. So if they fuse, they'll make a diploid cell. That will be 2n equals 10. And we'll have 5 pairs of homologous chromosomes. So let's draw out the pairs. Now they're not necessarily lined up like this in the cell, but I just want to show it this way so that you can tell there are 5 different pairs. So if this cell were to undergo meiosis, it's asking what is the probability that a gamete will have a set of chromosomes, all of which came from the original male gamete. So that will be all 5 of these blue chromosomes. Now for each chromosome, the cell has a half chance of giving the blue chromosome to its gamete. So let's draw another cell. So what we want is all blue chromosomes. Now here, it has a 1/2 chance of giving that blue one, and the same for the other, so let's just write 1/2 for all 5 chromosomes. So if you multiply that across, the probability of getting a blue chromosome for each pair of chromosomes, you get a 1 out of 32 chance that this will be your gamete, and that is the same as the original male parent here. Same set of chromosomes. So your answer is 1 out of 32.
Hello guys, it's Professor Schmidt. So let's do this problem, number 4. Indicate which stage of the cell cycle / mitosis the following illustrations represent. So when they word it this way, basically it means if the cell is in mitosis, what stage of the 4 stages, unless your class has gotten more specific about the stages, are we looking at? And if it's not in mitosis, then we're basically going to say that it's in interphase. So this shouldn't be so bad. Let's take a quick look. A here, excuse me, sorry about that. In A here, we have chromosomes that look like they're getting pulled up. I should say chromatids that look like they're getting pulled apart from one another. And that occurs during which phase? That occurs during anaphase of mitosis. So it's sort of taken for granted that if you say anaphase, you mean mitosis, but you might want to be more specific about that. The bottom line, you guys, is if you can see the chromosomes in this type of a situation, you're in mitosis. So we might as well just go ahead and say that this one where you just see the nucleus, you don't really see anything. It's chromatin that's very decondensed. That we're going to call interphase right there. So all the other ones are going to be phases of mitosis. So what about B? By the way, you can't tell from this picture, and I don't know why I wrote interphase is, you can't tell from this picture what part of interphase the cell is in, whether it's G1 or S or G2. There's not enough detail there. But all the rest of these must be phases of mitosis. So here we have doubled chromosomes, right? 4 of them, which looks like it's the diploid number for this cell. But they're not particularly arranged in any type of order, so that's going to tell us that it's prophase, right? When they're all lined up on the metaphase plate like this, then it's going to be metaphase, right? So lastly, we have pretty much 2 new cells, but we still see them. They haven't decondensed yet. That's going to be telophase or telophase. And that's really all they asked. If you wanted to, you could say that, okay, if you wanted to look at the order, this is number 1, this is number 2, that's number 3, and that's number 4, right? In terms of the prophase, metaphase, anaphase, telophase sequence of mitosis. Sometimes, you know, they don't ask you for these things, but it's not bad while we're doing it together to just mention it in case someone does ask you later. Okay, well that's pretty much it for this problem. In problem 5, we're asked a somatic cell at mitotic metaphase from a chimpanzee was examined and 96 sister chromatids were observed. What is the number of chromosomes that would be observed in a haploid gamete from this chimp? Okay, so the thing that's challenging about this question lies in the wording and your understanding of the difference between a chromatid and a chromosome. In metaphase of mitosis, remember, the cell has already gone through the S phase of interphase. In other words, the chromosomes have been doubled. So it just so happens, there's no way that you could know this unless you just happen to know it, but chimpanzees have a diploid number of 48. After they go through S phase, remember when one chromosome duplicates and becomes like that? These are two sister chromatids. There's a total now of 96 total chromatids. And whether you knew that there were 48 or not to begin with, you're given the information that there are 96 chromatids. I'm not sure I like the way they say sister chromatids there. Each one of them is a member of a sister chromatid pair, but the bottom line is that there's 96 chromatids there. If it's during mitosis, especially metaphase, because that's before they've separated, that number of chromatids that exist in that cell is going to be double the actual number of chromosomes that comprise the diploid number. All right? So what you really have to do here, they're not even, see, this is why it's a little sneaky. They're not even asking you what's the diploid number. But in order to know the haploid number, you've got to know the diploid number. So there's 96 chromatids, which translates into the diploid number being 48, which then translates into the fact that the haploid number is 24. And that's how many chromosomes you would find in a haploid gamete from a chimp. Most people know that, most of you guys know that humans have 46 chromosomes. We would have 23 in a haploid gamete, a sperm or an egg. And that also means that we would have 92 chromatids if we were observed one of our cells during metaphase of mitosis. Question 6 asks us, the number of chromosomes present in a muscle cell of a cat is 38. How many chromosomes would be found in a feline gamete? So the key to this question is to understand that in any somatic cell, they could have said here muscle cell, heart cell, brain cell, whatever it is, as long as it's not a sexual cell, then you're looking at the diploid 2n number, right? In a gamete, I think you guys know feline means cat, but cat gamete, simply you're going to have the n number of chromosomes. In this case, if 2n equals 38, then n is going to equal 19. And it's really as simple as that. All sorts of organisms have different numbers of chromosomes. If this was a human, we would have 46 chromosomes in our muscle cells, and we'd have 23 in a gamete. So just be careful about what type of cell, and I don't think most people expect you to know how many chromosomes a cat or a dog or a, whatever has. Probably good to know about a human. But the answer for this one is 19. So let's look at question 7 now. It's a bit of an odd one. It says assume the amount of DNA found in a somatic cell when it is in G1 is represented by the quantity Z. A, what is the amount of DNA found in a daughter cell after mitotic division? And B, what is the amount of DNA found in a cell during prophase? They don't say it, but prophase of mitosis, right? Now this quantity Z, this quantity Z, it could have been anything they want to say, an algebraic X, Y, Z, whatever. So Z is really not important here. That's just some unknown quantity. All you need to understand is the way the cell cycle and mitosis changes the net total amount of DNA. So before we even answer the question, let's just say this. They're saying it's a somatic cell in G1, right? So in G1, we have Z. And let's just for fun down here say, if it's a human, we have 46 chromosomes and 46 chromatids, right? We go through S phase. At the conclusion of S phase, what has happened? Well, we know that all the chromosomes have been doubled, so there are 92 chromatids. They didn't ask us this, but I just want to illustrate the point. That's double, right? So after S phase, I want to be very clear that it's after, you're going to have an amount that is 2Z, right? And then you're going to go through G2, where you still have, that may look like 27 here, but it's 2 times Z. You're still going to have 2 times Z. Then you're going to go through mitosis. What's the whole point of mitosis? Is to make 2 daughter cells that are back the same as the original, right? So after mitosis, you're going back now to having, well, let's just say Z in this cell and Z in that cell. If it was a human, you'd be back to 46. Remember, the whole point of S phase in duplicating the chromosomes is to have enough material to partition out to the 2 new daughter cells. So after doing that little analysis, we can answer very easily. After mitotic division, we have Z, the same, Z will just write. Looks like a 2, so I like to put that line in the middle of it. The same amount as a somatic cell when it's in G1, right? We see that in the drawing. During prophase in mitosis, before the 2 have split, though, we're still in the 2Z situation, right? So it's 2Z. Z is just, it could be X, like we said. It doesn't really matter. Don't get obsessed with the Z, the Z-ness about it, the whole thing. And by the way, I mean, you could say Z or you could say 1 times Z if you thought that was clear and, 1 or 2. But that's the idea of that one. So again, just be careful. The information they're telling you, somatic cell in G1, you have to be able to know how many chromosomes. I don't mean the exact number. I mean 1 complete diploid set is going to be there at that time. They could have given you information about a different time in the cell cycle, and that could change the question. But this is the way this one goes.